Hello to everyone watching this presentation. My name is Loïc Labache, and I am a postdoctoral researcher in the Department of Psychology at Yale. Today, I am going to talk about the atypical hemispheric specialization of the cerebral functional architecture. To get everyone on the same page, hemispheric specialization is defined as an interdependent relationship between a set of brain structures in one hemisphere and a given cognitive function. Hemispheric specialization is a fundamental principle found through the entire animal kingdom. In this work, we will engage hemispheric specialization through the perspective of language lateralization. So a prototypical hemispheric specialization at the population level is a left hemisphere dominant for language and the right one for attention. Therefore, and because we must start somewhere, a dominant left hemisphere for language will reflect a typical brain organization. The problem is that only few information is known on the relationship between all different lateralized cognitive functions. So to try to counteract this lack of information, I will first define the typical heteromodal language network atlas, and then I will use this atlas to identify typical and atypical individuals to extensively characterize them from a functional perspective. So first, I wanted to highlight the main language network. To do so, I took advantage of the Billie Jean database that contains the brain data of 144 participants who performed a resting state acquisition and 3 different language tasks. To identify higher order language area, I performed a conjunction analysis of the left activations and left asymmetries during the 3 language tasks. And this conjunction led us to select 32 supramodal regions of the left hemisphere involved in sentence processes. These regions are essentially located along the superior temporal sulcus to the angular gyrus and along the lower frontal sulcus too. But concerning the typical and atypical language organization, and as pointed out in the recent review by Vigerhertz, crucial information about the organization of hemispheric specialization in atypical individuals is missing. The information concerns, for example, the regional activation patterns of atypical individuals. The question now is why these atypical phenotypes have been so little studied to date. First, regarding language, because the probability of identifying an atypical individual in the general population is really low. And secondly, because patterns of hemispheric specialization in atypical individuals are highly variable. So to answer the question of characterizing the atypical organization of the language network, we will examine the functional organization of the language network in the dominant hemisphere, as well as its inter-hemispheric coordination with the mirror network in the opposite hemisphere. So I will now analyze the organization of the language network using 994 participants from the Human Connectome Project database. All these participants both completed a language comprehension task and a resting state fMRI acquisition. And using an unsupervised learning algorithm, I will investigate the language network organization at an individual level using 5 different biomarkers. First one, the functional asymmetries induced by the language task at the network and at the hub level. Next, the intrinsic homotopic interhemispheric connectivity, which represents the cooperation between the 2 hemispheres at rest. And finally, the strength sum and asymmetry of the intrahemispheric functional connectivity. And as you can see on the dendrogram, the algorithm allowed us to identify 3 groups of individuals, each having different language network organization. And I named the different groups of individuals according to the values of their asymmetry during the language task. I therefore have a first group of 480 individuals in orange that I named strong typical because they have strong left asymmetry during the language task. And the second group that I named mild Mid-typical in purple is also left asymmetrical but weaker. And finally, the last phenotype contains 82 participants, and all have a right asymmetry during the language task. And this group is, of course, a group of atypical individuals. But if I look more closely to the profile of each clusters, we can see that we reproduced our previous result published in eLife using a complete different database, showing that using multimodal markers allow to accurately identify atypical individuals. And once again, the identified atypical individuals do not have a brain organization mirroring that of typical individuals, with a language network underpinned by a left and a right network, both wired for language processing at rest. And now we have identified typical and atypical individuals, we can have a look at the relationship between atypical language lateralization and the more global macroscale organization of the cerebral cortex. So as you all know, the brain does not only support language processing. So the question that I will try to answer now is to what extent the lateralization of a specific function is reflected across the architecture of the sensory flow integration of the entire cortex as defined by Marcel Mesulam in 1998. To do so, I use the mathematical modeling proposed by Mark Gillis in 2016. It notably shows that using a diffusion map embedding technique, it is possible to obtain the functional topographic role of a brain region using the three first gradients. So I computed for every 995 participants, the three first functional gradient. And for remember, the first gradient is anchored on one hand in primary areas and in heteromodal association cortex in the other hand. The second gradient mainly opposes the primary visual cortex to the somatomotor and auditive one. And finally, the third gradient opposes the internal processes versus external ones. But because we want to assess the global brain architecture at the network level, I computed the average gradient value in each of the 7 large-scale resting state networks as defined by Thomas Yeo in 2011. That means that for each network, I have a gradient value in the left and in the right hemisphere. Next, I computed the asymmetry for each of the 7 networks and each participant. And finally, by merging the strong and mid-typical clusters, I investigated the impact of language phenotype on the 3 gradients asymmetry using a classical ANCOVA model. The results of the ANCOVA are really interesting because they first show that for the 3 gradients, the association cortex is the one with the strongest typicality effect, with asymmetries value significantly different between typical and atypical individuals. And the second interesting result from the ANCOVA is that the strongest dissimilarity between typical and atypical group is present in the 3rd gradient, with an opposite effect to the effect on the 1st and 2nd gradient. Indeed, here, atypical participants are leftward lateralized and typical ones are rightward lateralized. And finally, the question that we ask from here, and it's kind of a big question in the field, is to what extent these gradients that show different organization, depending of the language lateralization, are heritable. So to do so, I first conducted an heritability analysis directly on the language phenotype lateralization using a subset of 989 participants from the HCP database. And I found that language lateralization phenotype was 13% heritable. And next, I computed the same heritability analysis, but this time on the 3 gradient asymmetries for each of the 7 networks. And the result shows that the heteromodal associative cortex asymmetry was once again the most heritable, 19% on average. And once again, the 3rd gradient was the most heritable among the 3 gradients. So we can say that both language lateralization and gradient asymmetries are in part driven by genetic factors. So I think that you need to retain today that the presence of language network atypicality parallels the macroscale organization of the cortex spreading from the first gradient to the third one, and that the canonical language network appears to be one component in the broader hemispheric organization of the human brain, and it reflects the specialization of distributed large-scale network across the entire cortical sheet, and that both language and large-scale brain functional organization are highly heritable. And finally, these findings pave the way for a deeper understanding of the origins and relations linking population-level variability in hemispheric specialization and the functional organization of cortex. A lot of questions are still open, as for example, the exact role played by the corpus callosum in the entire hemispheric regulation, or the mechanism supporting the implementation of the asymmetry during development. And thank you all for your attention.